the University of Colorado Boulder. Those of us in astrobiology have been familiar with Carol's work for decades. She's been a regular at the astrobiology science conferences with the NASA Astrobiology Institute and so forth. She got her PhD at Brown, then did some postdoctoral work at Stanford before going to Colorado, where she's the director of the Center for Study of Origins and also a member of the Center for Astrobiology at Colorado. Her work has focused broadly in the philosophy of astrobiology. Most recently, and I think actually it's been a recurring theme, is this philosophical question of what is life? Right. She has a forthcoming book with Cambridge University Press on this subject. I've about halfway through it now, and it's really awesome. It's a very, very thought-provoking and profound work. And so it's my tremendous pleasure. I should also mention that Carol is a co-investigator with the Templeton grant that we have. George Cody and also Greta Heisted at Purdue are co-investigators, called Chance, Necessity, and the Origin of Life. So Carol's kind of gives us some philosophical grounding in that proposal. And today she's going to talk about searching for potentially biological anomalies using tentative criteria for life. Carol Cleland.
Thank you. Thank you. And it's a pleasure to be here, and thank you so much for inviting me to come. So what I want to address today is material there's a forum article in the journal Astrobiology that is coming out. It may already be out. It's fast-tracked, so it's on their website, which is actually a very similar title. I have it at the end. But anyhow, it's on this very topic that I'm going to be talking about. And Chapter 8 of my forthcoming book with Cambridge University Press's Astrobiology series, Chapter 8 is also on the material I'm going to be talking about today. And the way I got involved with this is I had written papers, some of you may have seen them, with Chris Chiba on why it's a mistake to define life, why you can't define life. And I've written a couple in addition to the ones I wrote with Chiba. And still, everybody wants to define life. NASA seems to think it can't go looking for life elsewhere unless it has a definition of life. The 2015 unpublished, it is unpublished, but the 2015 astrobiology strategy has a whole section devoted to key issues in defining life. The recent CRC handbook in astrobiology has a whole section devoted to definitions and the nature of life. So this obsession with definitions, I thought about it and I thought, well, I can understand this because how are you going to recognize it if you go out there and you find it unless you have a definition of life? And I thought more about this from a philosophical point of view, and the first part of the talk is going to be very philosophical, and I thought, well, you actually don't need a definition. This is a kind of red herring, this obsession with definitions. And so that's what this talk today is about. So to begin, there are three approaches to searching for extraterrestrial microbes, and it's microbes. I mean, everybody here knows that that's most likely what we'll find, and that makes the situation more difficult because obviously if you see little green men waving, on Mars, you're going to see that they're living things. But microbes you can't really detect with the naked eye, and you're going to have indirect tests for them, and so that makes it especially difficult for searching for life, particularly within our solar system. And we know that microbes are probably by far the most common form of life in the universe. So the most popular is defining life, and we have just oodles of definitions of life. And I want to say a few words about this, even though I'm not going to dwell on it, because I think it's really important to see why defining life is so problematic. Definitions are basically about language and concepts. When you define, for example, bachelor, the classic example in elementary logic books, what you do is you examine your concept of bachelor, and you dissect that concept into simpler, better understood components, being a bachelor, being unmarried, human, and male. So what you do when you give a definition is you dissect concepts. You don't look at the world. You're defining a word. And definitions, if you want to call something else a definition, it's not really a definition. That's what definitions have been. That's how logicians understand them, and definitions are about logic and language. So the problem with definitions is if you go to define life, you're going to be dissecting our current concept of life. And definitions work really well for human kinds, human categories, like garbage, bachelor, money, because there's no more to their nature than our concept. They wouldn't exist if there were no humans. If there were no humans, there wouldn't be money, there wouldn't be garbage. A lot of garbage is great for microbes. We call it garbage because in relation to ourselves. There wouldn't be bachelors, there wouldn't be widows. I could go on and on. But there are some common nouns, categories, that are not like this. And among those kinds of categories, we think of being water, heat, and water and heat and presumably life. Those would be examples. Quartz, although we've been having interesting discussions. Bob and I have been interesting discussions about quartz, but still there's something about the nature of quartz that allows us not to dissect our concepts, but to ask ourselves, gee, what really is quartz, as opposed to just what do I think it is, as a competent speaker of the English language. So the problem with definitions is that if you're talking about natural categories like water, quartz, presumably life, not necessarily, but presumably life, then dissecting your concept is only going to tell you what you think it is. It's not going to tell you what it really is. And the added problem we have is that we know that all life on Earth descends from a last universal common ancestor, and we know that there are biochemical alternatives, modest ones, that you could build perfectly functional proteins with different abiotic suites of amino acids. Yes, you have to make sure you have the right combinations of, of certain types of amino acids, but nonetheless, people have constructed proteins, and protein chemists in the laboratory that would be functional in the right organismal environment. Similarly with nucleic acids. We know there are at least 12 bases that could function in duplex DNA. We know there are possible alternative sugars. So we know there are alternative possibilities, and we don't know how different life could be from life as we know it. So we have reasons to believe that we're dealing with a single example of life on Earth, and even worse, and I can't go into this in detail, but 2 chapters of my book are devoted to this topic. Most of our theorizing in biology about life is based on plants and animals. It goes back 2,000 years. People still think about organisms in the terms of plants and animals. And you really see this with talk about evolution, where people kind of try to fit microbes, which engage in lateral gene transfer, which makes them, it's very difficult to construct a phylogenetic tree for them, into the sort of nice, neat categories that true multicellular eukaryotes fit into, which gives you a fairly natural tree of life. So we have reasons to believe that our current thinking about life is based on an unrepresented example of Earth life, primarily, and moreover, that familiar Earth life itself may be unrepresentative. It's certainly a single example. That means we should be really worried about thinking that if we define life in terms of dissecting our concept of life, that it's going to be very useful for looking for novel forms of life on other worlds. That's all I'm going to say about definitions. I swore I'd say less, but I happen to really think it's important to understanding why definitions are just such a red herring, that they're more likely to result in the classification of truly novel life descended from an alternative origin as non-living than they are to have it classified as living. So definitions are a problem. Preliminary universal theory of life, well, it's implicit in contemporary practice, but it really doesn't exist. Biologists are really fond of saying, and I've had Norm Pay say this to me many times, give me a biological principle and I'll show you an exception. I mean, that's his favorite response to talking about any fundamental principles of biology. And if you look at fundamental principles of biology, that seems to be true. And people love in biology coming up with them. And philosophers of biology have become pluralist as a result. That is to say, they have suggested that really there isn't, there's never going to be a fundamental theory of life. Life just does not have natural categories. But I think this is all premature if we're dealing with an unrepresentative subsample of a single example of life, namely familiar Earth life. So having said that, I want to now focus on searching for potentially biological anomalies. That's my proposal. My proposal is forget hunting for life per se. Let's focus on finding potentially biological anomalies, and instead of using defining criteria, use tentative criteria. So I'm going to develop that idea in this talk. So sorry to have gone on for so long, but I think it's really important to see why this is a reasonable proposal. So the dilemma is, how can we recognize truly novel forms of life, examples of life as we don't know it, if we don't have a definition or universal theory of life? It's not at all obvious that we will recognize it if we see it, especially if it's microbial. And there are two strategies, basically, for searching for extraterrestrial life. One is I call searching under the street life for life like ours. And that is definition-based. That is, design equipment and methods for detecting specific kinds of acceptable deviations from familiar life. Think about what, look for different abiotic amino acids. Look for, collections of R amino acids instead of L amino acids. These are the kind of acceptable deviations that we might be able to recognize. Whether they exist is another question. The other possibility is searching for anomalies, physical chemical systems that resemble familiar life in provocative ways, and yet also differ from it in baffling ways. And so my recommendation is that we don't search under the street light. That is to say, the street light is like the definition. It's showing you only what you're looking for. And what I'm suggesting is, if we run into life on Titan, that's my favorite spot, that's really different from familiar life, then most likely you're not going to find it because you'll be looking for the wrong thing. What we want to do is recognize those cases that have the most promise for being a novel, unfamiliar form of life. And this may be true in Mars, too. So I want to talk now philosophically about Thomas Kuhn's work. I don't agree with his conclusions, but I think there was an important insight in Thomas Kuhn. And it was this notion of a paradigm. He argued that normal scientific research, which is what all you are doing, with a few exceptions perhaps, is conducted within the confines of paradigms, like the Dana system of minerals, which include not only theories, but also methods, instrumentation, classic experiments. Kuhn threw all kinds of stuff into the paradigm, and they're just the set of things that you train your graduate students and your postdocs are the right way to look at a certain kind of problem or domain of study. And that's what he argues. And what's really interesting is that paradigms are both friend and foe. They're friend in the sense they're invaluable tools for scientific research, facilitating the construction of hypotheses, design of experiments, and interpretation of results. They can nevertheless hinder the exploration of nature, discouraging certain avenues of research and biasing the ways in which data are interpreted. And if you went with the definition of life, this is exactly what you're doing. You've got a paradigm. And you're going to be basically not recognizing stuff that doesn't fit it, and it amounts to not recognizing anything that isn't terribly, really a lot like familiar life. So let's talk more about paradigms. So violations of a paradigm, according to Kuhn, are initially seen as mere puzzles that can be solved within the paradigm given significant ingenuity. So he argued that, look, when you have a paradigm, any thing that looks like a counterexample to that paradigm is going to be seen as just a puzzle. You just have to be clever enough to use the tools. Remember all the different tools in the paradigm? Let's see quickly. All of these things are tools that you can use to solve the puzzle. And so you assume it's solvable as a puzzle. But he argued that under the right circumstances, a puzzle becomes viewed as something more serious, an anomaly, vis-a-vis the dominant paradigm. And when something becomes seen as an anomaly, the paradigm structure is automatically being challenged. Now, Kuhn argued, so Kuhn argued that anomalies pave the way to scientific discoveries because they weaken the grip of a scientific paradigm on the scientific mind. But he also argued that you couldn't recognize something as an anomaly until you actually had a crisis or you were posed for a scientific revolution. So there was this kind of the catch-22, that is to say, if everything seems like a puzzle until the discipline is thrust into a crisis where you're posed on a scientific revolution, then it seems like how are you ever going to break out of the paradigm? It's a huge problem for Kuhn. Philosophers have recognized it for a long time. And that's why Kuhn is not widely accepted by philosophers of science anymore. This is basically his picture. You have normal science, and you have a puzzle, and the puzzle You go back and forth like this until the puzzle begins seen as potentially anomalous, which leads to a crisis, which may go back to it being a puzzle and a revolution. And he argued quite notoriously that political, cultural, and psychological circumstances were required for a revolution. What I want to do is short-circuit this ridiculous process. What I want to argue is that we can recognize anomalies, we can actually look for them, rather than just assuming they're puzzles and we're not clever enough to solve them. And I want to give you some examples of anomalies to bring that out. This is the famous example from Kuhn that he cites as an example of how hard it is to see a puzzle as anomalous. So William Herschel, before William Herschel, from 1782 to 1792, some of the best observers in Europe had observed a star in the position we now know is occupied by Uranus. Herschel had a new instrument, a very fancy instrument of his own design, which was more powerful, a telescope. And he looked in the same spot these other people had looked and was struck that the object was not very star-like. It was disc-shaped and it moved among versus with stars. He would have taken time to do this. He had to track it, but he noticed that this was an anomaly. And he wrote a paper, which was distributed over Europe, saying that the new object was a comet. Up until this time, there were thought to be no planets, basically beyond Saturn. So this was considered to be impossible, but everybody agreed there were comets and there were planets. So what Herschel did is he viewed it as a puzzle within the dominant paradigm, and he identified it as a comet. Well, people all over Europe with the same inferior telescopes where they'd previously seen a star, some of the best observers in Europe suddenly saw that it was anomalous. Before, they hadn't seen it as being anything odd. Now they saw it as anomalous, and the very famous Anders Lexell, within a few months, pointed out that the orbit was not, in fact, cometary, it was planetary. And within a very short time, 6 months after this, suddenly everybody was seeing planetesimals, mostly asteroids, everywhere, where they hadn't been seen before with the same instruments by which they had previously been studying the stars. So what's really interesting about this is that it exhibits what Kuhn calls the discovery of, basically he argues that there was a modest paradigm shift with the discovery that Uranus was not a star but a planet. And Kuhn, he was a physicist and a philosopher and a historian, and he focused primarily, because he was a physicist, on cases from the physics. He had lots and lots of cases from physics and chemistry, the physical sciences. But it turns out there are lots of them in biology, and they exhibit the same kind of trait. Consider, for example, Tom Cech's discovery, Nobel Prize winning, Cech's discovery of catalytic RNA, which challenged a certain version of the central dogma of molecular biology that Crick had stated, holding that there is a rigid functional division between nucleic acids and proteins. Proteins supply the catalytic material for life. Nucleic acids cannot play the role of biocatalysts. And as many of you probably know, maybe you don't know, Cech didn't initially conclude when he had what he found. So he was investigating splicing, which was a very poorly understood phenomena at the time. And he had what he thought was a pure sample, a precursor RNA that didn't have any protein. He had, basically synthesized it himself. And he observed, he and his team, splicing activity going on. And he didn't understand how this could happen because there were no protein catalysts, and only proteins could catalyze the splicing of these precursor RNA molecules together. So he's deeply puzzled, but he didn't conclude, oh, I've got catalytic RNA. No, it took him more than a year. He and his team struggled to figure out where the mysterious protein was. He assumed, because this was the paradigm, that it must be a protein catalyst that was responsible for the splicing. And it's really lovely. I'm going to show you some quotes from his Nobel Prize lecture, which really brings this out. He called it the experiment that never worked. He discovered catalytic activity, splicing, and the preparation of RNA, which they thought they'd removed all protein. They tried every way they could think of to remove proteins. They heated it up. They used, to denature proteins. They They used proteases to denature them, and it failed. It took them a year before they finally accepted, even considered the idea that RNA must be catalyzing itself. Here's the quote. I love this quote directly from his Nobel Lecture. Once you give Nobel Lectures are wonderful sources for philosophers and historians, because once you win the Nobel Prize, you're not embarrassed anymore. And Tom Cech concedes he was so embarrassed by the results that he snuck into the lab at night to figure out what was going wrong. He was just concerned he'd be embarrassed and there was some simple mistake here with his postdocs. So he goes, our first hypothesis was a splicing activity was a protein tightly bound, perhaps even covalently bonded. That's pretty outrageous, right? To the pre-RNA isolated from the tetrahymena nuclei. This would have been a very unusual protein-RNA complex to survive multiple forms of abuse to which we had subjected it. That we took this hypothesis seriously, this is the important part, provides an indication how deeply we were steeped in the prevailing wisdom that only proteins were capable of highly efficient and specific biological catalysis. Again, here's a case where the dominant paradigm is turning what is actually an anomaly, sign that there's a problem with a major scientific principle into a mere puzzle to be solved. And they treated it as a puzzle. It took them over a year before they returned to it, and they said, well, look, maybe it's the RNA. This is pretty outrageous, but maybe it's the RNA. Discovery of viruses, same phenomena. Challenged assumptions about the size of viruses. Initially, it was classified as bacteria on the basis of their very large size and gram-positive staining. It took 10 years to get them classified as a virus. They were as big as some of the parasitic bacteria that are found. So they thought, well, these must be bacteria. They were actually found in a cooling tower when they were investigating, what's it called? I've just gone blank on the disease. They found them in amoeba, and they were just little spots. And they were really shocked. Actually, they weren't initially not even shocked. They were just a puzzle, you know. These were awfully big, awfully small bacteria, but they must be bacteria because they stained gram-positive, and viruses are very small. And there was this view that viruses were small because they didn't need all of this equipment, to function, since they parasitize or they invade cells to reproduce. So that was the idea here. They didn't need ribosomes and things like that. So they could be small. And so nobody expected ever to see a virus this big. And so it took 10 years before these were actually recognized as viruses. And right away... How big was that? Huh? When you say so big, how big is that? I don't have that. I meant to write that down, and I don't have it. So I'd have to look it up, but I'd be happy. It's in my book and my papers, okay? So I can't remember how big it is. But it was bigger. It was this... Pardon? Yeah, it was actually, they were as big as certain, what were well known to be parasitic bacteria in certain cells. So they were, in certain organisms. So it wasn't hard to have them classified this way. But what's really striking is once they were identified, and it was actually, I think it was 1992, but I have 93 here, but they were reclassified as a virus in 2003, immediately, even bigger viruses were found, including Pandora virus, and it's visible under the light microscope. And so there's also another even bigger virus that has recently been found. But it's just like the sudden discovery of many different planetesimals after the discovery of Uranus. They were all there all along, but they were unrecognized for what they represent. And that's true here, too. They were just presumably being seen and classified as bacteria. So discovery of Archaea is another example. Before the discovery, so the discovery of Archaea challenged the distinction between prokaryotes and eukaryotes, which by the middle of the 20th century had been thought to be evolutionarily significant for perfectly reasonable reasons. That is to say, the distinction was based on the morphological characteristics of certain unicellular organisms. Some of them have membrane-enclosed nuclei, other ones don't. And so it seemed perfectly reasonable to think that this was a fundamental evolutionary difference. And these morphological characteristics, which gave you for single-celled organism, a distinction between two different kinds, prokaryotes and eukaryotes, was thought to be significant phylogenetically. And when Carl Woese and Sidney Fox were analyzing the genomes of various bacteria and Archaea, they discovered something truly astonishing, that there were sequences of that they were, in their terms of their genetic mechanism, some of these Monera, that's what they were all classified as Monera, and then unicellular eukaryotes were protists, ignoring fungi for the minute, because they're not relevant to this discussion. But they discovered that the Monera included these organisms that had very different genetic apparatus, and the genetic apparatus of some of them was more similar to that of protists than to other Monera, which was problematic. And then they discovered the lipids in the membranes of the same ones that were more similar to protists were actually more different than both protists and bacteria, what we now call bacteria, other Monera. And the result of this was they proposed completely redoing the kingdoms of life. And finally, the suggestion was, they originally had Archaea bacteria. Finally, the suggestion, as everybody knows, was 3 domains and 6 kingdoms. And now there is certain debate about whether or not all of these, the eukarya, should be placed under the Archaea. But I don't want to go into that now, except to mention that there was tremendous resistance to Woese's suggestion. He received a great deal of disparaging comments. The great Ernst Mayr said very nasty things about it. And it took, several dozen years before people were willing to accept that actually the Monera were not a natural phylogenetic category. And here's a lovely quote. This was actually, there's plenty of evidence before this time that this was the case, that people should have been suspicious, but they were, again, they were mesmerized by this paradigm, the prokaryote eukaryote paradigm. And here's a lovely quote, 1978, about the time all this was happening, but before Woese was accepted. The fact that these two bacteria, they're actually Archaea, have similar lipids is of interest, but almost certainly this can be explained by convergent evolution. This hypothesis is strengthened by the fact that Heliobacterium, another Archaean, quite different organism, also has lipids similar to the two acidophilic thermophiles. Sorry about my pronunciations here. So the point here is this was viewed as puzzling but easily explicable. And this is typical of what happens to a phenomenon that doesn't quite fit a paradigm. It's seen as a puzzle, but not as threatening to the paradigm. And so the role of anomalies in scientific revolutions, I think Kuhn is right about this. He said the role of anomalies is that in most cases when you have major scientific revolutions, it is preceded by a puzzle, what was seen as a puzzle, sometimes for dozens, even 100 years, as a puzzle becomes seen as an anomaly. And all the cases I just mentioned involve phenomena that, while present all along, were unrecognized for what they represent because they conflicted with assumptions or categories provided by a reigning scientific paradigm. They were either not detected because they weren't looked for, or they were detected but misidentified because they violated prevailing assumptions. So Kuhn thinks this is kind of a hopeless situation. What I'm going to suggest is that, no, it's not hopeless. You just have to look for the anomalies. And sometimes, most of the time, they will just be puzzles and you can solve them. But sometimes, some of them will be anomalous enough that they're deserving of further scientific research for their potential to really change our ideas about the phenomena concerned. So searching for potentially biological anomalies using tentative versus defining criteria for life. Potentially biological anomalies are physical systems that defy classification as life or non-life. They straddle the borderland between life and non-life. And because they do this, they're most likely going to be declared non-living, because definitions give you a thumbs up or a thumbs down. They supply necessary and sufficient conditions and completely limit the category of items concerned. So our definition of bachelor, if something is unmarried human and male, adult they usually throw in, then it's a bachelor. If it's a bachelor, it's unmarried human and male. End of story. But what you find with natural categories that we're trying to explain scientifically is often you will find these exceptions, these outliers, these borderline cases are the especially interesting ones. And what you want to do then is not just dismiss them as non-living, and I'm going to give you an example in a minute of where that happened, but you want to recognize them as potentially important examples, as potentially biological anomalies in the case that we're discussing here. And what are tentative criteria for life? They include both allegedly fundamental and non-fundamental, universal and contingent characteristics of familiar life, which are deployed jointly for the purpose of detecting potentially biological anomalies. And I want to say just a little more about what I mean about this. So instead of looking for life with a favored definition, like the chemical Darwinian definition, or like, and I'm just going to talk about it in a minute, the metabolic chemical definition that was used in the Viking missions, go with both. Why have to sit there and decide between rival definitions? Rather, look for both kinds of characteristics. And there's a vast difference between going from a so-called general definition to actually translating it into search criteria. That's another issue. You aren't just going to look for metabolism that's chemical. You're going to look for specific sorts of things. We're going to talk about that in a concrete example in a minute with the Viking missions, which look for the ability to metabolize a carbohydrate solution. And also look for contingent characteristics that are you don't think are fundamental, but are universal. I'm going to give you a list in a minute, which will bring this out more clearly. And look for things which are uncommon among living things, but really rare among non-living things. So I'm going to talk about some of this at the end of the talk, if I have time. But the important thing is tentative criteria are not defining. That is to say, they're meant only to show you that a system, and you're going to have to make your mind up about whether this system, a particular system is like this, is worthy of further investigation for life. When identified, an anomalous physical system becomes a focus for further scientific investigation. This contrasts with definitional and preliminary theory approaches, which are focused on finding life per se, and identify cases that do not conform to the set of privileged criteria in use as non-living, period. And here's the hard part. And I actually hoped originally to come up with an algorithm. And then I realized as I struggled, I talked to statistician friends, formal epistemologists in philosophy, and we tried hard to come up with an algorithm. And suddenly it struck me in the middle of all this that of course you can't find an algorithm. Why? Because you can't have an algorithm for looking for the unanticipated. How can you have an algorithm for looking for something you know not what? It's only intelligent creatures like us who can say, that shouldn't be there. That's weird. But you don't have an algorithm for looking for weird things because they're going to be too weird. So I gave up on that project. I have a degree in math, and I was really excited about it. I was really disappointed to give up on it. So if somebody could talk me into going back to it, I'd be happy to do that. So it's going to take human decisions about whether a system is anomalous enough to be worthy of scientific investigation. But we humans are really good at detecting anomalies as long as we're aware that they might exist. And the key is viewing them not immediately as puzzles and persisting in viewing those puzzles to be solved for years sometimes, but immediately saying, this could be an anomaly, maybe it's a puzzle, let's explore it a little further. So I want to just go back to the Viking missions briefly. Because I think they're a really classic example of an anomaly. And I think they're an example of anomaly for 40 years that people have been treating as a puzzle. And so I want to talk about that. So the Viking label release, I'm going to focus on for this discussion. And the PIs, Levin and Stratt, of the LR experiment concluded, yes, they found life because the Martian soil released radioactively labeled carbon dioxide gas when inoculated with a radioactively labeled nutrient solution. And Martian soil heated to a temperature of 160 degrees for 3 hours, a temperature high enough to kill any known bacteria on Earth, failed to release radioactively labeled CO2. So the important point here is that it behaved just as expected of bacteria in pre-mission tests. They did pre-mission tests, And this is what they expected to find if there was life on Mars. They got that. But things became weird after that. But let's look at Levin's words. The Viking missions labeled release life detection experiment returned data from Mars fully satisfying the pre-mission criteria for the detection of microbial metabolism. So he emphasized the pre-mission criteria, and he's thinking of this in terms of defining criteria, not tentative criteria. If he were thinking in terms of tentative criteria, he wouldn't have been so quick. NASA concluded no, it's very interesting, on the following empirical grounds. No organic compounds were found in Martian soil analyzed by the Viking GCMS. The GCMS was in fact elevated to a fourth experiment, which it wasn't originally intended to. The LR response from Mars was thought to be too much too soon compared to Earth microbes. I love this one because it looked like the graph. If you look at the graph of the reaction when it was exposed to nutrient solution, Earth microbes were much slower and the Martian microbes were much faster. So what they did is take what initially looked like a metabolic criterion and they made it even more Earth-centric. Does everybody see that? More Earth-centric. So their definition, which was broader, became narrower to reject the thought that they had found life. Excuse me. A second injection of LR nutrient, this was, I think, a key thing. So this was probably the thing that actually made the difference. But we now know, by the way, there are organic compounds have been found on Mars. I don't know whether you're aware of that, but I'm sure you are, actually. Sorry. But they have found organic compounds on Mars. A second injection of LR nutrient onto positive soil samples not only failed to reinvigorate the evolution of carbon dioxide, as usually occurs with microbial-containing Earth soils when the little buggers become hungry, but also about 30%, this is weird, of the gas evolved after the first injection was reabsorbed into the soil. So these are the grounds that NASA gave for no. This is probably the heavy that made their decision. What happened then was efforts, this was NASA's conclusion was accepted, and efforts were made to find an inorganic compound that could explain all of these phenomena here, which had not been anticipated. Various abiological candidates for the Martian oxidant have been explored over the years, including hydrogen peroxide, superoxides, exotic oxidation states of iron, and most recently, perchlorate. Now, whatever you think of perchlorate doesn't, if you think that perchlorate's it, I don't want to get into that discussion, except to notice that perchlorate was many years afterwards. And so they were looking for an inorganic oxidant, and maybe it was a puzzle. Maybe it was perchlorate. But the important thing, that it was a potentially biological anomaly still remained, even if it was perchlorate. And it should have been, no, it was never investigated as actually the possibility of a biological oxidant. It was always assumed there must be an inorganic oxidant, and we have to be clever enough to find it. Now, from what I've read, I'm not convinced it was perchlorate, but it doesn't matter for the purposes of my argument, because the point is that they kept looking for an inorganic oxidant. They never considered the possibility that it was organic oxidant. They never had a mission go back to look for an organic oxidant. And of course, I love this owl oxidant. It's a band, actually. Yet life is also an oxidant. That's the important thing. Truth is, neither side of the debate over the LR experiment knew what they found. The results of the LR experiment were potentially biological anomalous, and had they been treated as such, both possibilities, biotic and abiotic, would have been seriously entertained and explored. So that's the difference between a definitional approach where it's thumbs up or thumbs down. In this case, it was thumbs down. Everybody's looking for an inorganic oxidant, and treating it as a potentially biological anomaly, which may turn out to be a puzzle, but is worthy of looking for the possibility of life, a biological oxidant, as well as inorganic oxidants. And I just want to list, I talk about these extensively in my papers and my book about the sorts of characteristics that you might use in as tentative criteria in a collection of criteria, because you're never going to discover an anomaly with just one criterion. You need to have a set of criteria, and what you do is you look for characteristics in this set of criteria, giving you a profile that looks anomalous. That is to say, you get enough pings on the things that look biological, but also some that look completely not biological, unexpected, unanticipated if it's a living thing, and you then are going to have to make the decision about whether or not it's worthy of further investigation for an unfamiliar form of life. And some big differences between definitions. This is a major one I mentioned earlier. Allegedly fundamental or defining characteristics should be deployed together instead of treated as rivals. Universal characteristic of familiar life, they're not thought to be fundamental life. For example, nucleic acids or complex polymers. Maybe nucleic acid, you might go looking for nucleobases, you might look for, proteins, amino acids, you might look for abiotic amino acids, but you're going to be looking for things that you think of as universal to Earth life, but you aren't convinced are universal to all life. And then contingent non-universal ways in which organisms found certain types of environments modify them, sedimentary structures, reactive gases, like methane and oxygen, which sometimes can be explained abiotically, but are often a sign of biological activity. The banded iron formations on Earth, for example. Characteristics rare among living systems, but even rarer in non-living systems, such as magnetite crystals. I think that, that in the case of the Allen Hills meteorite, we now know that you can make, or at least it has been argued that you can, by Golden and others, that you can make similarly chemically pure and structurally perfect, geometrically perfect, tiny magnetite crystals abiologically. But the fact that we didn't know that that could be done at the time is enough to have meant that it was anomalous. So the sort of having to decide immediately, biology or no biology, I guess, biology or no biology, that's too hasty. My suggestion is that you collect this stuff and then you look and see whether or not you've got a system that you've got a number of these things that look really lifelike and a number of others that don't look very lifelike, and then you proceed to design a search strategy for potentially biological anomalies. And here are the papers. So the Moving Beyond Definitions and the Search for Extraterrestrial Life is coming out in Astrobiology. This issue, it's already fast-tracked. And then my book, which should be in print in October, chapter 9. So I've covered a lot of material in a really fast fashion, so I would love to have questions. It's hard to, I could just talk could be 4 hours long, obviously, right? So I tried to give you the big picture, and I'm hoping you ask me some great questions. Thank you.
The questions, please, Larry.
So you mentioned in passing Titan and said it's one of maybe what you think is most interesting, but then you didn't mention it.
Because I think it would, if there's life on Titan, it's not going to be much like our life.
Right. So what kind of experiment would you propose doing on, say, a Titan lander for anomalies?
So I think Chris McKay's his Lego block. Okay. Oh, okay. So he argues that what's really interesting about familiar molecules is that they have a kind of periodicity and a small number of types of molecules that appear in a certain structural pattern. And he's thinking of amino acids, complex polymers with a certain repeating segments of certain types of molecules. So he proposes that we look for that. So we wouldn't be looking just for carbon molecules, or we wouldn't be looking just for amino acids, but we'd be looking for these kinds of phenomena. Unfortunately, I'm not a scientist, so I have the great privilege of just suggesting to you to look for things without knowing how to do this. But I think, you look for thermodynamic, you look for basically systems that are that have thermodynamic properties that shouldn't be there given the environment they're in. You would look for signs. So one of the hard problems with definitions that people gloss over is that you have to translate a definition, like a metabolic definition, into actual things to look for. And so the way they did that in the LR experiment was they had a nutrient solution, ironically designed for most organisms to be cultivated in Earth. They weren't aware that less than 1% of Earth microbes can be cultivated. So that was a significant problem they weren't aware of. But it was a perfectly reasonable approach to kind of make concrete this abstract notion of biological metabolism. There are real problems with the chemical Darwinian definition, because how are you going to be able to detect Darwinian evolution in a small enough a frame of time for an experiment to run. There have been some interesting suggestions about how to do that. So I think what you have to do is pay a lot of attention to what the characteristics are, kind of in a general sense, and then how you translate those into criteria. There's a difference between criteria, right, and a characteristic. Criteria are search for a characteristic. So, and I think looking for complex structures that shouldn't be there. I mentioned sedimentary structures. If you're on Mars, it would be really interesting to look for past life, including signs of things like stromatolites or oxidized minerals that probably Mars never got that far along to have, I don't know, but it's fairly recent on Earth. So, but anyhow, you're going to look for stuff that shouldn't be there, that is biologically suggestive, but you're not going to be discouraged or give up because it has some features that seem to be inconsistent with life as we know it. That's the main lesson from this. And you want a set of criteria, because only with a set of criteria is it going to pop up as anomalous in a really robust sense. I hope that helps.
So Carol, I've got a question about this distinction between puzzle
Yes.
and anomaly. And I think you kind of alluded to this, that there's somewhat of a continuum, and some things that there's easier and harder puzzles, and harder puzzles can grade into easier anomalies and harder anomalies. Is there some, I mean, we talked about defining terms and classification. Yeah. Is there a classification of anomaly versus puzzle?
No, the problem is, and here's where I think Kuhn was right. I mean, Kuhn says if you have a paradigm, a scientific theory, and all the stuff that goes with it, you never just have a theory alone. You have certain instruments. For example, it's really interesting in quantum chemistry versus quantum physics. The instruments that are considered acceptable for running tests are quite different. So you have, everybody, both quantum chemistry and quantum physics accepts quantum theory, but they have all of these special methods and instruments unique to their own disciplines that are designed to, detect the things that they want to detect. So when you have a paradigm that gives you methods, it gives you instrumentation, it gives you theory, including subsidiary assumptions, it's very easy to see a counterexample as merely a puzzle that we haven't been clever enough to solve. Now, let me just give you some other examples of truly cases where you have puzzles that were actually solved. So black holes were predicted long before they were discovered. They were predicted as a consequence of Einstein's theory of general relativity back when it was first proposed, almost, just within a very short time of its proposal. But they weren't discovered until many, many dozens of years later. Certainly the Bose-Einstein condensate, I don't know how many of you know, it's a state of matter. It's considered to be a completely different phase where you get down within a fraction of a degree of 0 degrees Kelvin, and suddenly a bunch of rubidium atoms begin behaving as if they're a single atom. And it was predicted from quantum theory dozens of years before anybody was clever enough to form it. And that was Carl Wyman and Eric Cornell at the University of Colorado Boulder is the one who finally was figured out how to get close enough to zero. Because, of course, you have to pump out all this heat, and pumping generates heat. It was a real puzzle, close enough to zero to actually form the Bose-Einstein condensate. So those are cases where the inability to find black holes and the inability to create the Bose-Einstein condensate were not viewed as challenging the theory. They were just puzzles that people hadn't been clever enough yet to solve. And so the problem becomes, when do you view something as a puzzle and you pursue it long enough, it suddenly becomes to be considered an anomaly. And I don't think there is an algorithm for this. I think it relies on scientific ingenuity and a deep understanding that we've tried a lot of things. It's like Tom Cech when he was trying to remove protein catalysts. And basically, he tried just about everything that was reasonable. Boiling them, using proteases, I can't remember what else, but he didn't even then conclude maybe the RNA is doing the catalysis. He didn't conclude that. They just put it away in exasperation for a year and then came back to it. So I don't know the history of Sidney Altman's. They shared the Nobel Prize, how his particular work worked, but I do know that in case of Tom Check, he just couldn't believe it. And so, to make a long story short, I think you should be ready to believe, or you don't have to say it's an anomaly, you have to say it's a potential anomaly. And you try to solve it as a puzzle, but if it resists being solved for long enough, then I think you should reasonably consider that you've got a genuine anomaly, and you might begin thinking through your theoretical structure that's basically it's so inconsistent with. Does that, I hope, helps?
I have two things. One is I'm an amateur mushroom guy, mycology. And what has happened there is over the years, everything was done by morphology. This mushroom looks like this, its color is different, but the shape is the same. Well, now with RNA studies, no, these guys are completely different.
Excellent,
This
yeah.
one isn't this, it's not a
Yes.
Russell, it belongs way over here with Anomalies.
Yeah.
It's just really crazy what that's done. And that was a paradigm that was there for.
And it's a perfectly reasonable paradigm because, before you understood the genetics of life, the genetic basis of life, morphology seemed like a good criterion. It was, by the way, the criterion that led so many people to believe that all the species were created separately before Darwin, because they were all so different morphologically. Birds didn't look anything like, let's say, humans and squirrels don't look anything alike, so they must be separately created. How could they have a common ancestor? And so it's a perfectly reasonable assumption until you begin doing what Darwin did and you look at the fossils of various organisms. Or you go to, and you notice that the finches, they're all finches. He was so worried about whether they were all finches that he, in his travels, that he actually went to, took them back and asked one of his ornithological comrades to confirm that they were, because they looked so different. Their beaks were different shapes. They had very different environments. And, this guy said, yeah, these are really weird looking finches, but they're all finches. So the point I'm trying to make is that Darwin's travels and his study of many different kinds of organisms led him to see that there were a lot of anomalies in the paradigm at the time that all of the species were created separately.
The other one is a researcher or processor induced anomaly. And this goes back about 30 years. Started me thinking that in looking at the data, and this was data for the weather models, I think it was temperature data, I can't remember, but there was an algorithm would throw out sort of anomalous values that were thought to be anomalous.
That's
Sort of
another
high
point.
temperature value.
Yeah.
So we're going to throw it away because it doesn't fit
Excellent
what we
example.
think about.
Yes.
And
That's a paradigm.
Then what happened is, as they were doing the climate data sets, they went and they found this and they looked at it, and it changed what the perspective was for the climate data set.
Right.
And that was just because they thought this is anomalous, so we'll throw it away.
Or an outlier. I wouldn't say, because the way you're using the word anomaly is not the same way I'm using it. Yeah, but outliers. I mean, people tend to throw out outliers, and it's reasonable. But sometimes those outliers are really important, and they're actually anomalies in my sense, not just outliers, which are just kind of noise in the data, right?
So I've got a question about defining what a paradigm and a paradigm shift is. There are clearly some ideas in science that were wrong. The Ptolemaic model of the cosmos with Earth at the center and the sun going around, that was displaced. You could argue that Copernicus came with a paradigm shift that could be changed our worldview. But you could also argue that Check's work on RNA, the fact that it had catalytic properties was just sort of, there are lots of chemicals that have catalytic properties, and they were thinking of proteins as the most likely source, but if proteins don't work, then acids being catalytic and lipids can be catalytic and RNA can be catalytic. And so is that a paradigm shift, or is it just a puzzle that took them a year to figure out? And I say this partly because I know that in science it's really popular to say, oh, this person made a paradigm shift. And we all want to think that we're doing, but most science, most of the time, are not paradigm shifts. And I'm not sure if I've seen one other than
So,
plate tectonics in our field in a very long time.
so there's Kunz View, and then there's my view. I mean, most philosophers of science don't accept Kuhn anymore. He's very popular among scientists, or was at least 20 years ago. But he has this very precise story about, I think I can bring it back here, which I put up here just quickly. I didn't go into a terrible detail, but Kuhn has a very precise story about how scientific change, revolutions occur. Oh, there we are. Sorry, there it is. And that's his story. I don't accept that story. So my answer to you is that the idea of a paradigm shift, I think that there's no crisp line saying that you've got a paradigm and then a paradigm shift and a revolution. For him, a paradigm shift is you completely have got a revolution. But Kuhn himself had trouble with that. So he talked about minor paradigm shifts. Like he uses the example of Herschel's discovery of Uranus, and he talks about it being a minor paradigm shift related, he claims, to the Copernican revolution, but he doesn't, let's leave that aside, because people then thought there were no planets beyond Saturn, that anything else, we knew all the planets, and anything else was either a comet or a planet. And so basically, that's why he came up with it being a comet. And so the paradigm shift for him was basically that, actually, I think it was not planet, it was a star or comet. So I've got that wrong, actually, star or comet. And so planet wasn't in the running for him as a possibility. So that's the paradigm. But that's a really minor paradigm. And the Crick's version of the central dogma of molecular biology was just what I said, that only proteins could be biological catalysts. So, what I think that was the paradigm, and I think Kuhn would have been happy with calling that a paradigm. That he was, but I think in terms of paradigm shifts in the robust Kuhnian sense, he himself waffled, which is why Kuhn is not widely accepted among philosophers of science. But I think the important lesson to learn is that your commitments, whether or not they're big theoretical ones, are just general assumptions that you make, that you work with, which are not always embedded in the theory, but are really widely held, like the central dogma of molecular biology, or like there are no planets beyond Saturn. I think that can completely determine what you consider a puzzle. And it's very hard to go from a puzzle to anomaly unless you're open to the possibility. And I'm asking people to be open to puzzles being anomalies.
Well, if there are not any more, one more, Tim.
I was wondering if you have any practical advice within the like on a life detection mission. You have some constraints with the weight and the mass of what you bring, and you want to do the experiment, like for example, your Mars, you want to do the inorganic experiment or the biological experiment. How do you decide sort of what instrumentation you would bring?
So that's an interesting question. I spell out this in much greater detail in chapter 8 of my book. And it's a little bit spelled out in this, the astrobiology article. But of course, I couldn't go into detail. Sarah Johnson at Georgetown and I are getting together. And she wants to take these ideas and work with me and her team to see if we can make them more concrete, to do exactly what you're talking about. Because she's interested in looking sort of beyond the box. So I'm hoping that will be something that will come out of this. We've just sort of been talking. I think we're going to be meeting at AbSciCon, and then she wants to bring me out to DC for talking with her team. So this is a possibility. All I can do as a philosopher so far, without talking to scientists, because this has to be a collaborative effort, is talk about the sorts of things that might be useful in finding something that looks like it shouldn't be there, that's potentially biological. That's the key. It looks like it shouldn't be there. Lots of things are going to look like they shouldn't be there, but they're not going to look potentially biological. So the key is identifying when it looks potentially biological, as well as, gee, that shouldn't be there. And that's hard. But I think you have to use a suite of criteria, and I think they have to be a broad suite, and I think they have to be tailored for particular planetary environments. That is to say, I think it's Sarah Seeger who says, that life is planet specific. And I think that's right. I mean, I think that you don't expect, if that's why I mentioned Titan. If there's life on Titan, it's not going to, it's definitely, if there's life on Europa, which probably has water, liquid water like our water, it might be quite a bit like ours. I don't know. Same with Mars. Early Mars was a bit like our climate. But if it's on Titan, that's why I like Titan, it can't be like ours. So it's going to be really different. That's why I think Titan's a place to look for really weird life.
Let's thank Carol again, and she'll be around this afternoon if you'd like to. Thank you.